to talk to you guys about human genetics. In the last lecture, we talked about Mendelian genetics, or inheritance, and now we're going to talk about human genetics, which is not nearly as straightforward as humans are, much more complex, I should say, than pea plants, as you could probably imagine. So when we start to think about human genetics, what we're really trying to kind of understand is how things are passed down from generation to generation, as you guys all know. And we're going to focus on a few genetic disorders because that's one of the ways that we have kind of gained some understanding about human genetics. A genetic disorder is any disease that's caused by an inherited mutation, essentially, that's passed from a parent to a child. And by studying some of these genetic disorders, it's given a lot of insight into how things are inherited, and that's kind of the topic of our discussion today. So in order to understand human genetics, there's some tools that are sort of helpful, one of which is a pedigree. So a pedigree is kind of like it's a chart that's very similar to a family tree, and it's basically showing genetic relationships between family members, and it can be mapped across generations. So pedigrees give us an opportunity to analyze information in a way that allows us to learn about inheritance of particular disorders. So in this slide on the screen here, there are two different pedigrees. The one on the top is a pedigree looking at a family with a condition called polydactyly. And the one on the bottom is looking at families with a condition called albinism or albinos. So on the top, let's just look quickly at a pedigree. So it looks like a family tree. And just to orient you, first of all, we're looking at generations here. So here's 1 generation, 2 generations, 3 generations, and you can see those marked by the Roman numerals on the side there. In pedigrees, females are represented by circles and males are represented by squares. A line that connects the circle and the square here is indicating that these two have reproduced, and the line that comes off then attaches their children. So these two people have one child, one male child, one female child, another female child, another male child, and another female child. So that is 5 children. And when the circle or square is shaded in, like you see up here on number 2, this male right here, that means that they are affected with the disorder. In this case, the disorder is polydactyly. So we have these two people. The male has polydactyly, and then when we look at their children, of which they have 5, 1, 2, 3 of their 5 children are affected. One of the males, number 1, and number 5 and 7, those are females, they all have polydactyly. Now, you see that number 1 here, this male that has polydactyly, he reproduced with this female, and they had 3 children, and one of their 3 children, one male, has polydactyly. These two have not reproduced, but we used to call this the marriage line, but I try not to say that anymore because that doesn't really mean a whole lot in this day and age. However, these two are united by some bond, and they have not yet had any children. This number 7, this female, has reproduced with number 8, this male. She has polydactyly, and one of their 2 children, the female, has polydactyly. So it's just showing us that we have 3 generations of people. The disease that we're looking, the genetic disorder that we're looking at is polydactyly, and it's represented in all 3 of these generations. And in a little while, I'll give you some questions to ask when you're trying to figure out the mode of inheritance, one of which is the condition that you're looking at, is there evidence of it in every generation, or does it skip generations? And the other question to ask is, are males and females affected more or less equally? So the answer to those 2 questions would be question number 1, does it skip generations? No, it's evident in all 3 of the generations that are mapped out on our pedigree, and males and females are both affected relatively equally. So if we go down to the next pedigree on the bottom, looking at albinism, so in this case, we have a situation where we have the circles or squares halfway shaded. And if the circle or square is halfway shaded, that means that that particular individual is a carrier, but they don't actually present the disease. So it means that they have a gene, one allele for it, but they don't have both alleles for it. So in most cases, those conditions are recessive. In all cases, I should say, those conditions are recessive. And the only way to show the trait of, in this case, albinism, would be if you inherited 2 recessive alleles for that condition. Remember, an allele from the previous lecture, an allele is just an alternate form of a gene. So for every gene or every trait, we have 2 alleles. And we could have 2 dominant alleles, we could have 2 recessive alleles, in which case we would call that homozygous dominant for the dominant alleles, homozygous recessive for the recessive alleles, or heterozygous for one of each. So in this case, the carriers are heterozygotes, meaning they have a dominant gene and they carry the recessive gene. But in order to present, to be an albino, you have to have inherited 2 recessive genes for that condition. So let's look at our pedigree here. So we have a female carrier and a male who is unaffected. They have 2 children. Their female child is a carrier. Over here we have another couple. The male in this case is a carrier, and they have 3 children. Their their son is also a carrier. Their son marries this family's daughter, both of them being carriers, and 2 of their 3 children are albinos. So these 2 children, the first male and the third female, or second female, pardon me, those 2 people have inherited 2 genes for that. So they have the 2 recessive genes, and that's perfectly possible because we know that if you cross 2 heterozygotes, your chances are getting one of 4 of them would be homozygous recessive. So the odds are that they're going to have at least a couple of these children, and they have 2 of their 3 are affected. So the question here would be, so these people don't have it, right? They're just carriers. So the condition does skip generations, right? We have 2 generations before we actually see the disorder. But males and females in our representation here are affected equally. Okay, so what all that means is, of course, we know that in humans we have 23 pairs of chromosomes, 23 homologous chromosomes, for a total of 46 chromosomes. And we inherit 2 alleles for each gene, right? And genes, of course, are located on our chromosomes. If the genes are located on chromosomes numbers 1 through 22, those are called autosomes. If the genes are located on the sex chromosomes, pair number 23, those are sex chromosomes. So, as a general rule of thumb, if the trait is carried on the autosomal chromosomes, numbers 1 through 22, historically, the condition will be present in females just as often as it is in males. So, again, the genotypes, we write the 2 alleles. So, for example, a big A, little a would be, in this case, a heterozygous configuration. And if that particular configuration was found on any one of the first 22 chromosomes, we would call that an autosomal, a gene that's found on an autosomal chromosome. Now, if the trait is carried on the sex chromosomes, it's going to be carried almost always on the X chromosome. There's not a lot of information on the Y chromosome. So the sex chromosomes are chromosomes number 23, as I said a minute ago. And of course, the sex chromosomes are the chromosomes that determine sex, which is why they're called the sex chromosomes. All the other ones are the autosomes, and they determine all the other stuff. The sex chromosomes carry other information other than just sex, though, of course. And so there's some of these disorders that are carried on these sex chromosomes. the autosomes, the homologous chromosomes, I should say, of the autosomes are all alike in terms of length, shape. So number one's the same length as the other number one. The shape and the genes that they carry, the pairs are all identical. Well, they're not identical because the alleles can be different, but they carry the same information. And then the sex chromosomes are assigned letter names. The X chromosome, the options are X or Y's. So in humans, males have one X chromosome and one Y chromosome, whereas females have two X chromosomes. The Y chromosome turns out to be a lot smaller. I don't know that I have a picture. I think I do have a picture coming up in a moment. But if you think back to the karyotype that we saw in the previous lecture, the sex chromosomes were a lot smaller, and the Y chromosome was really small. So because human females have two copies of the X chromosome, all of the eggs that they produce are going to contain one X chromosome, and the X chromosome is going to be passed. That one X chromosome will be passed to their offspring. Males, on the other hand, have an X chromosome and a Y chromosome. So the odds are that half of their gametes will contain X chromosomes and the other half are going to contain Y chromosomes. Again, that's just an odds. It could be a little bit different, but that's the odds. A flip of a coin kind of a thing, 50-50 chance. So if we have a trait, an inheritable trait that's carried on the X chromosome, it's much more likely to be seen in men than it is in women, because men only have one X chromosome. So if it's on the X, then they don't have any opportunity to hide it behind the other X, right? They only have one X. And so if it's on that chromosome that they inherit, then they are going to show the condition. Okay. So how we write the genotypes for the X chromosomes, you can see on the slide there. For a female, you would have big X and then your letter for your allele. In this case, we're looking at that heterozygous presentation. So big X, big X, little a for the female. For the male, they only have one X, so they have big X. In this case, the big A is the one they inherited, right? A male can inherit one of these two, right? So half of these are going to be in some of the gametes, the other half are going to be in the other gametes. So depending on which egg gets fertilized, the male is going to get one of these two configurations. In this particular case, they got the big X, the big A, and then the Y chromosome. So if this X is carrying some kind of abnormality, it's going to show up in this guy. Whereas in this one, it could potentially be hidden depending on the situation. All right, so let's look at some more examples. So when we go back to the autosomal traits carried on chromosomes 1 through 22, oftentimes the question is going to be, is this an autosomal dominant type of inheritance or an autosomal recessive? And that's where the does it show up in every generation question comes in. So if the condition shows up in every generation, then chances are that's an autosomal dominant condition. If it skips generations, then chances are that's an autosomal recessive condition. So autosomal dominant conditions are more rare. And the reason for that is because you can't hide. It can't hide. If it's autosomal dominant, all you need is one dominant gene to present the condition. And so oftentimes these dominant disorders produce very serious side effects that show up pretty much right at birth. And so the person with that particular allele doesn't oftentimes live long enough to reproduce, although there are some exceptions to that I'll mention in a little while. So sometimes we do see cases of autosomal dominant genetic disorders that are produced by a new mutation, right? It just shows up, for example, in a generation. But that's sort of outside of this discussion right now. So if the condition does not skip generations and it shows up equally in men and women, we're going to guess that that's an autosomal dominant condition. Now, if it does skip generations and it doesn't show up for a while, and it also affects men and women equally, then we're going to think of that as an autosomal recessive disorder. These are much more common. And again, how we kind of can guess that is because that's where these carriers come in, right? We wouldn't have known. If you just saw these two people, you don't know that they carry albinism. You didn't know that these people carried albinism. You don't know until they have children with it. And so when it shows up, then you start kind of working backwards. And that's kind of how pedigrees work. You start with what you know, and then you kind of work backwards to sort of fill in the rest. Now, X-linked recessive disorders are disorders that are carried on the X chromosome, and they are only evident if the person has the recessive alleles. So for a female to express an X-linked recessive disorder, she has to have inherited 2 recessive alleles. So both of her X's have to carry that recessive allele. So it's not as common in women. However, for men, all they have to inherit is 1 recessive allele on their 1 X chromosome. They don't have another X, so they don't have an opportunity to hide that allele behind a dominant gene or a dominant allele. So because men only have 1 X chromosome, they're going to express any allele, any X allele, whether it's dominant or recessive. And if that recessive one is carrying a trait, if they inherit a recessive one and it's responsible for a recessive disorder, then they're going to exhibit that disorder. But for a female to exhibit that same X-linked recessive disorder, she's going to have to inherit 2 alleles, recessive alleles. So both of her parents would have had to have had the recessive allele in order for her to show it. So X-linked disorders are not as often evident in females as they are in males. So again, here's a picture of an X chromosome and the little Y chromosome, and here's 2 X's up here. So we write our genes, the sex chromosomes, we write the genes as X's and Y's, and then as a superscript, we put our alleles, right? So gene B on the X is going to control your ability to distinguish between red and green. So this is a color blindness. This is a very common X-linked recessive disorder. So the options are the big B if they can see, if they're not colorblind, if they can distinguish between red and green, and a little b if they cannot. So we would then, if we were trying to figure out if this person was going to be colorblind or not, we could do a Punnett square based on what we know about their parents. The gene for colorblindness does not exist on the Y chromosome. It's exclusively on the X. All right, so take a minute to try to draw out a Punnett square for this situation where we have big B being our full color vision, little b being your red-green colorblind. So you're going to try to draw a Punnett square for a cross between a colorblind female, so a colorblind female, and a male that has full color vision. So a colorblind female. And remember, this is X-linked recessive. So take a moment to do that. Okay, so hopefully you had a minute to do that. So let's see how you did. So we're going to do our Punnett square with a colorblind female. So a colorblind female, in order for the female to be colorblind, this is a X-linked recessive. So for our female to be colorblind, she has to be X little b, X little b. Those are her alleles. With a male with full color vision. So on his X, he's got a big B, he is not colorblind, and he has a Y. So hopefully your Punnett square kind of looks like this. So we've got X big B, X little b. This would be a female who is not colorblind, because for her to be colorblind, she has to inherit both little b's. X little b from mom, Y. So we've got a male who is colorblind. Our other box here, X big B, X little b, another female who is not colorblind, and another colorblind male. So we have a colorblind mother and a man with full color vision, and none of their females are going to be colorblind, but all their males will be colorblind. And the reason for that is because it's on the X. She only has colorblind genes to give. All of her male children are going to be colorblind. Period. Females, not as much. If she mated with a colorblind male, then all her female children would be colorblind as well. But only, that would be the only scenario. So hopefully you got that. If you didn't, I'd stop now and think about it for a minute. Okay, so here's a big pedigree of an X-linked or sex-linked trait. And how we know that is because just by looking at this, right, we see, first of all, look, there's only squares. Only males have this condition. That's a big tip-off that this is a sex-linked disorder. And it is skipping generations. We don't see it here, we don't see it here, and then we see it here, and then we start to see more and more and more. Part of this is because this was a very shallow gene pool, right? These royal people were really into reproducing with a small group of other royal people. Didn't work out so well because it turns out that they carried this X-linked recessive gene, which predisposes them to hemophilia, which is a bleeding disorder, and that wasn't working out so well, especially affecting the males, because if you remember from your history, the males at this time, the males were the kind of the jewels of the family, so to speak. So their males were getting less and less robust as time went on, as you can see, and they were afflicted with this bleeding disorder, which is oftentimes not compatible with life, right? They can bleed out very easily with not a whole lot of trauma. All right, so we don't really know. So if you looked at this, you'd be like, well, where's this coming from? Who can we blame for this? Well, we don't really know given this information, but we can start to kind of put it together if we sort of zoom in on it. So let's zoom in on it a little bit. So here we are. We're going to look at this section. So I'll go back and show you where we are. So here we are right here, right? So we're going to look at between Alice. Alice is daughter of Leopold, Duke of Albany. Alice's son Rupert has hemophilia. Turns out Leopold had it as well. So let's look here. So Leopold, we know, her father had hemophilia. She does not have it, but her son has it. So remember, this is sex-linked recessive. So what do we think Alice's genotype is? So just some test-taking strategy. It's X-linked, so we know we can cross these two off right off the bat. We're going to write these on X's. So it was one of these two. We know Alice does not have it, but her son has it. So she has to have one recessive trait for it, but not two. If she had two recessive traits for it, she would be a hemophiliac. If she had no recessive traits for it, her son would not be a hemophiliac. He had to get this from her, and she has to carry it. So that gives us answer number 4 is the correct answer. Big X, big A, big X, little a. So we could go back in here and we could put her in as a carrier, right? And so you can kind of do the same sort of thing here. You can figure out who's carriers and who's not based on what, working backwards from these people that are affected. So like, for example, if we come over here, we see there's 3 of these sons, 2 of them have it. They had to get it from someplace. She has to be a carrier, right? We're guessing, we're not sure where there's the question marks. We're not really sure, right? We don't have quite enough information there. We don't know if Alice's daughter might be a carrier. She might be, she might not be. We don't know yet because none of these kids, we don't have enough information at this point. We know he doesn't have it because it's X-linked. All the males, they either have it or they don't. So none of the squares are carriers. They can't be. They either have it because it's on the X chromosome or they don't. But we're not so sure about the circles. They may be carriers, they may not be carriers. If their brothers have it, then that means their mother carries it. So there's a chance that these women are carriers, but we don't know for sure. We won't know until they start having children. If they have children and their sons have it, then you know that they are carriers. So it's kind of it's kind of complicated, but sort of interesting at the same time. All right, let's think about this a little bit more. So again, determining our mode of inheritance by looking at our pedigree. So remember I said earlier that there's two questions that we want to be able to answer. One is, are there about the same number of men and women? About, it doesn't have to be exact, but they're equal, they're, you know, more or less equally represented. That would tell us that this is a, if the answer to that is yes, males and female are more or less equally represented, that would be autosomal. If men have it more than women, then it's X-linked. And then does the trait ever skip generations? If it does, it's if it does skip generations, chances are it's a recessive. And if it doesn't skip generations, it's probably dominant. So if it skips generations and it's affecting men more than women, like the one we just saw, then that would be sex-linked recessive. If it has men more than women, but it doesn't skip generations, then we would say sex-linked dominant. Are you getting the hang of it? Okay, let's look at some more. So here is a condition called a hypertrichosis, which is a real thing, which causes a lot of hair growth, as you can see. And we're looking at our pedigree and we're answering our two questions. Is our first question is, are men and women equally affected? More or less. The answer to this would be yes. You see squares and circles that are filled in. And then the next question is, does it skip generations? So the answer to that is no. So men and women are equally affected means it's autosomal. Skipping generations, the answer to that is no. That would be autosomal dominant. Here's another one. This is a condition called androgen insensitivity syndrome. And androgen insensitivity syndrome is a situation where there is a loss of sensitivity to androgens, which are sex hormones. So the loss of sensitivity to these people, they have a genetic mutation that makes them unable to respond normally to male hormones such as testosterone, which is an androgen. It can also be caused by a deletion on a gene called the S as in Sam, R as in Ron, Y gene, SRY gene. It's a pretty critical gene. It doesn't act alone, but it's found in both males and females. But it directly influences the development of secondary sexual characteristics that ultimately distinguish men from women. So when it's absent, those genes produce female characteristics, and when it's present, it produces male characteristics. But with individuals that have androgen insensitivity syndromes, the males are female in appearance, and they can even feel female even though they have an X and a Y chromosome. So it's kind of an interesting situation. So it's sex-linked, obviously, and it's skipping generations. So it is sex-linked recessive. So hopefully that's starting to make a little bit of sense to you guys. This is another condition. This is an autosomal dominant condition because it does not skip generations and it affects men and women equally. This is what a pedigree for Huntington's disease look like. If you remember from when I was talking earlier, autosomal dominant conditions are not very common because they tend to not be compatible with life in most cases. So people die before they reproduce, but Huntington's disease is an exception to that because the symptoms sort of stay hidden until the person gets to like their late 30s, early 40s, in which case that's when they start to see symptoms. And for many people, they've already reproduced. So again, that's sort of the beauty of the karyotype, and the benefit of the karyotype is you can start to kind of map these diseases, right? Because a disease like this, it causes all sorts of issues, uncontrolled movements, loss of the mental faculties, caused mostly by brain cells that die off, but it doesn't show up usually until somebody's 40s, and they can carry the allele and reproduce before they know that they have it. So if you have a family history of Huntington's disease, that would be something you would probably want to think seriously about before you reproduce, or do some screening of the fetus to make sure to check to see if that gene is present. Okay. So actually I'm going to stop this video here, and I'll just for the purpose of time, and I'll pick up the next one.